Anatomy and Physiology, Chapter 3, Part 3, The Clinical Connection. Digitalis often is given to patients with heart failure, a condition of weakened pumping action by the heart. Digitalis exerts its effect by slowing the action of the sodium-potassium pumps, which lets more sodium accumulate inside the heart muscle cells. The result is a decreased sodium concentration gradient across the plasma membrane, which causes sodium-calcium antiporters to slow down. As a result, more sodium remains inside the heart muscle cells. The slight increase in the level of calcium in the systole of heart muscle cells increases the force of the contraction and thus strengthens the force of the heartbeat. Transport in vesicles. A vesicle, as noted earlier, is a small spherical sac. As you will learn later in this chapter, a variety of substances are transported in vesicles from one structure to another within cells. Vesicles are also important materials for Vesicles also import materials from and release material into extracellular fluid. During endocytosis, materials move into a cell in a vesicle formed from the plasma membrane. In exocytosis, materials move out of a cell by the fusion with plasma membrane of vesicles formed inside the cell. Both endocytosis and exocytosis require energy supplied by ATP, thus transport in vesicles is an active process. Endocytosis. Here we consider three types of endocytosis: receptor-mediated endocytosis, phagocytosis, and bulk phase endocytosis. Receptor-mediated endocytosis is a highly selective type of endocytosis by which cells take up specific ligands. Recall that ligands are molecules that bind to specific receptors. A vesicle forms after a receptor protein in the plasma membrane recognizes and binds to a particular particle in the extracellular fluid. For instance, cells take up cholesterol containing low-density lipoproteins, LDLs, transferrin, an iron-transporting protein in the blood, some vitamins, antibodies, and certain hormones by receptor-mediated endocytosis. Receptor-mediated endocytosis of LDLs and other ligands occurs as follows. Binding. On the cellular side of the plasma membrane, an LDL particle that contains cholesterol binds to a specific receptor in the plasmic membrane to form a receptor LDL complex. The receptors are integral membrane proteins that are concentrated in regions of the plasma membrane called clathrin-coated pits. Here, a protein called clathrin attaches to the membrane of its cytoplasmic side. Many clathrin molecules come together, forming a basket-like structure around the receptor LDL complexes that cause the membrane to invaginate, fold inwards. Again, many clathrin molecules come together, forming a basket-like structure around the receptor LDL complexes that cause the membrane to invaginate, fold inward. Vesicle formation. The invaginated edges of the membrane around the clathrin-coated pit fuse, and a small piece of the membrane pinches off. The resulting vesicle, known as a clathrin-coated vesicle, contains the receptor LDL complexes. Uncoating. Almost immediately after it is formed, the clathrin-coated vesicles loses its clathrin coat to become an uncoated vesicle. Clathrin molecules either return to the inner surface of the plasma membrane or help form coats on the other vesicles inside the cells. Fusion in endosome. Once again, fusion with endosome. The uncoated vesicle quickly fuses with a vesicle known as an endosome. Within an endosome, the LDL particles separate from the receptors. Recycling of receptors to plasma membrane. Most of the receptors accumulated in elongated protrusions of the endosome. Most of the receptors accumulate in elongated protrusions of the endosome. These pinch off, forming transport vesicles that return the receptors to the plasma membrane. An LDL receptor is returned to the plasma membrane about 10 minutes after it enters a cell. Degeneration in lysosomes. Other transport vesicles which contain the LDL particles butt off the endosome and soon fuse with a lysosome. Lysosomes contain many digestive enzymes. Certain enzymes break down the large protein and lipid molecules of the LDL particle into amino acids, fatty acids, and cholesterol. These similar molecules then leave the lysosome. The cell uses cholesterol for rebuilding its membrane and for the synthesis of steroids such as estrogen. Fatty acids and amino acids can be used for ATP production or to build other molecules needed by the cell. The clinical connection. Viruses and receptor-mediated endocytosis. Although receptor-mediated endocytosis normally imports needed materials, some viruses are able to use this mechanism to enter and infect body cells. For example, the human immunodeficiency virus, HIV, which causes acquired immunodeficiency syndrome, AIDS, can attach to a receptor called CD4. This receptor is present in the plasma membrane of white blood cells called helper T cells. After binding to CD4, HIV enters the helper T cell via receptor-mediated endocytosis. Phagocytosis. In high school, excuse me for this footnote, in high school I learned that this term was phagocytosis. In college I learned that it was phagocytosis. Aluminum, aluminium. I don't know, you pick. Textbook says phago, to eat. Phagocytosis is a form of endocytosis in which the cell engulfs large solid particles such as worn-out cells, whole bacteria, or viruses. Only a few body cells, termed phagocytes, are able to carry out phagocytosis. The two main types of phagocytosis are macrophages, located in many body tissues, and neutrophils, a type of white blood cell. Phagocytosis begins when the particle binds to a plasma membrane receptor on the phagocyte, causing it to extend pseudopods. Projections of its plasma membrane in cytoplasm. Pseudopods surround the particle outside the cell, and membranes fuse to form a vesicle called a phagosome or phagosome, which enters the cytoplasm. The phagosome fuses with one or more lysosomes, and lysosomal enzymes break down the ingested material. In most cases, an undigested material in the phagosome remain indefinitely in a vesicle called a residual body. The process of phagocytosis is a vital defense mechanism that helps protect the body from disease. Through phagocytosis, macrophages dispose of invading microbes in billions of aged, worn-out red blood cells every day. Neutrophils also help rid the body of invading microbes. Pus is a mixture of dead neutrophils, macrophages, and tissue cells and fluid in an infected wound. So it's very interesting in that an invading body is approached by a phagocyte, engulfed, dissolved, and gotten rid of. There are a lot of video presentations. There's a lot of little snippets out there. And with this book, I got this book when I went to school over at Middlesex Community College. With this book comes a card. This card. And it gives you access to the Wiley's Plus site. So, on that site, there are many videos. There's many different kinds of media. And this is what I was saying earlier. Now, this book, this narration, can be completely useful to someone who without sight, who has no sight, or somebody who's in their car, or somebody who just wants to hear the material again and again and again. I'm that type of person. I need to hear it again and again and again. I need to read it. I need to reread it. I need to see it in several different forms of media to retain the information. And over at Wiley's, where the book comes from, they know that. And so when you go to the back of the book, or you get this card and you approach them and you sign up for your account, and you can access all of these films and videos and presentations and animations and experiments and everything you can see. It makes it more interesting than just words on a page. It makes it more easy to digest. Phagocyte makes it more easy to digest than simply reading it. And it is a great tool for all of those returning students who are my age or long before the internet and all of this terrific media that we have today existed. We sat there with books and we tried to hash it out, but now things are so much easier and so much more interesting. All those thoughts that we had about something, a material or a subject is difficult or it's boring, all of that is dispelled by the fact that we have several different ways to digest the material. Now, all that aside, I want to go back to my original thought. I really do seem like I'm shucking the book to you, but I find it's very interesting. It's interesting, super complicated, but once you start to grasp it and you go back to every single word that you don't understand. If you take a piece of paper with you, no matter what the subject matter is, take your notebook and write down, you're scrolling down a page. You're reading the page and you come to a word that you don't understand. The general rule of thought is this: if you come to a word on a page that you don't understand and you blast by it as if the word doesn't exist, you will not understand that entire page. You have to understand every single word that is written to digest all of the material present. It seems like it would be common sense, but it's not. Take that piece of paper, write things down. Now, to go back to what I was originally saying, phagocytosis, one of the most amazing things that I saw through this site, I don't know if you can just go out on YouTube and find it. You might be able to, or I know that you can find it on their site, but it was a microscopic illustration. So it was actually a video. I believe it was a captured video, microscopic video. Now, I said illustration, which would mean someone drew it, but no, I believe it was an actual cell that they recorded via microscope, and it was a phagocyte. And it is amazing. Earlier in the videos before this, I described the human body as a symphony of organisms that live together to form our human body. We work together to a goal. And I hope to get into psychology, a psychology book that I have, and to narrate that and describe in detail the id and the ego, the superego, and the various components of the mind that come together to make your daily decisions. But one of the most interesting videos by far that I came across was a phagocyte. And speaking of a symphony of organisms, was this organism that is part of your immune system. And the video opens with a phagocyte coming across the page, coming across the screen. And if this thing didn't look like an octopus, that was a squid. That was an octopus, I'm telling you. And it came across an invader. It worked its way towards it. It engulfed it. And then it secreted its ink, lysozyme, and that's how it does it. It just engulfed it, secretes a substance, lysozyme, which is an enzyme that just dissolves the invading body, and now your immune system has worked for you. Completely amazing, and it just really ties everything together. In the oceans, you look and we see these large macro organisms. That is a squid. That is an octopus, and it's like, we have that on board. That's one of my relatives right there. Very interesting. So if you're in this class, and you're using this as study material, it would be worth it to go to the back of the book. And try to find these videos that exist. So now here we are, finishing up phagocytosis, transport across the plasma membrane. Most body cells carry out bulk phase endocytosis, also called pinocytosis. A form of endocytosis is in which tiny droplets of extracellular fluid are taken up. No receptor proteins are involved. All solutes dissolve in the extracellular fluid, are brought into the cell. No receptor proteins are involved. All solutes dissolved in the extracellular fluid are brought into the cell. During bulk phase endocytosis, the plasma membrane folds inward and forms a vesicle containing a droplet of extracellular fluid. The vesicle detaches or pinches off from the plasma membrane and enters the cystole. Within the cell, the vesicle fuses with a lysosome, where enzymes degrade the engulfed solutes. The resulting smaller molecules, such as amino acids and fatty acids, leave the lysosome to be used elsewhere in the cell. Bulk phase endocytosis occurs in most cells, especially absorptive cells in the intestines and the kidney. Exocytosis. In contrast with endocytosis, which brings materials into a cell, exocytosis releases material from a cell. All cells carry out exocytosis, but it is especially important in two types of cell: secretory cells that liberate digestive enzymes, hormones, mucus, or other secretions; nerve cells that release substances called neurotransmitters. In some cases, wastes are also released by exocytosis. During exocytosis, membranes enclose vesicles called secretory vesicles form inside the cell, fuse with plasma membrane, and release their contents into the extracellular fluid. Segments of the plasma membrane lost through endocytosis are recovered or recycled by exocytosis. The balance between endocytosis and exocytosis keeps the surface area of a cell's plasma membrane relatively constant. Membrane exchange is quite extensive in certain cells. In your pancreas, for example, the cells that secrete digestive enzymes can recycle an amount of plasma membrane equal to the cell's entire surface area in 90 minutes. Transcytosis. Let me correct that. Transcytosis. Transcytosis. Transport in vesicles may also be used to successively move a substance into, across, and out of a cell. In this active process called transcytosis, vesicles undergo endocytosis on one side of a cell, move across the cell, and then undergo exocytosis on the opposite side. As the vesicles fuse with the plasma membrane, the vesicle's contents are released into the extracellular fluid. Transcytosis occurs most often across the endothelial cells that line blood vessels and is a means for material to move between blood plasma and interstitial fluid. For instance, when a woman is pregnant, some of the antibodies across the placenta into the fetal circulation via transcytosis. Once again, for instance, when a woman is pregnant, some of her antibodies cross the placenta into the fetal circulation via transcytosis. I'm going to read a table which will summarize the various types of transport. Then, in the next segment, we will cover cytoplasm. Transport of materials into and out of the cells. Transport process. Passive processes. Movement of substances down a concentration gradient until equilibrium is reached. Do not require cellular energy in the form of ATP. Diffusion. The movement of molecules or ions down a concentration gradient due to their kinetic energy until they reach equilibrium. Simple diffusion. Passive movement of a substance down its concentration gradient through the lipid bilayer of the plasma membrane without help of membrane transport proteins. Nonpolar hydrophobic solutes, oxygen, carbon dioxide, and nitrogen gases, fatty acids, steroids, and fat-soluble vitamins move by simple diffusion. Polar molecules such as water, urea, and small alcohols do as well. Facilitated diffusion. Passive movements of a substance down its concentration gradient through the lipid bilayer by a transmembrane protein that functions as chemical carriers or channels. Substances that transport in this manner are polar or charged solutes, glucose, fructose, galactose, some vitamins and ions such as calcium, chloride, sodium, and calcium. That would be potassium, chloride, sodium, and calcium. Osmosis, the passive movement of water molecules across a selectively permeable barrier from an area of higher water concentration to an area of lower water concentration. Active processes, movement of substances against a concentration gradient, require cellular energy in the form of ATP. Active transport. active processes in which a cell expends energy to move a substance across the membrane against its concentration gradient by transmembrane proteins that function as carriers. Polar or charged solutes use active transport. Primary active transport, active processes in which a substance moves across the membrane against its concentration gradient by pumps that use energy supplied by hydrolysis of ATP, sodium, potassium, calcium, hydrogen, iodine, and chloride. Secondary active transport, coupled active transport are two substances across the membrane using energy supplied by a sodium or hydrogen concentration gradient maintained by primary active transport pumps. Antiporters move sodium or hydrogen and another substance in opposite directions across the membrane. Symporters move sodium or hydrogen and other substances in the same direction across the membrane. Antiporters move calcium, hydrogen out of cells. Symporters move glucose, amino acids into the cells. Transport in vesicles. Active process in which substances move into or out of cells in vesicles that bud from a plasma membrane. It requires energy supplied by ATP. Endocytosis. Movement of substances into a cell in vesicles. Ligand receptor complexes trigger infolding of a clathrin-coated pit that form a vesicle containing ligands. That is, receptor-mediated endocytosis. Ligands, transferrin, low-density lipoproteins, LDLs, some vitamins, certain hormones, and antibodies travel through receptor-mediated endocytosis. Phagocytosis, cells eating. Movement of a solid particle into a cell after pseudopods engulf it to form phagosomes. Bacteria, viruses, and aged or dead cells, like blood cells. Phagocytosis was just discussed. Bulk phase endocytosis, cells drinking. Movement of extracellular fluid into a cell by infolding of plasma membrane to form a vesicle. Solutes in extracellular fluid. Solutes in extracellular fluid travel by bulk phase endocytosis. Exocytosis, movement of substances out of a cell in secretory vesicles that fuse with the plasma membrane and release their contents into the extracellular fluid. Neurotransmitters, hormones, and digestive enzymes travel in this way. Transcytosis, movement of a substance through a cell as a result of endocytosis on one side and exocytosis on the opposite side. Substances such as antibodies across endothelial cells. This is a common route for substances to pass between blood, plasma, and interstitial fluid. Next, we will discuss cytoplasm. And the objective of this section will be to describe the structure and function of cytoplasm. No, that's not it. The objective of this section will be to describe the structure and function of cytoplasm, cytosol, and organelle.